Hi everyone. This is the second lecture for your chapter cell cycle and cell division. In the last class, we discussed what is a cell cycle, how much time it takes, what is interphase, and what follows interphase, which is mitosis or meiosis. We studied mitosis, which is again divided into two parts, cytokinesis and karyokinesis. In karyokinesis, we studied the four phases, which were prophase, metaphase, anaphase, and telophase. After which, we also studied cytokinesis, how the plant cell divides and how an animal cell divides. We finished off with discussing what the function of this cell division or mitosis is. In today's class, we will be discussing meiosis, which is synthesis of haploid cells. Okay, meiosis is happening or it occurs to help in reproduction. Okay, it is the first step in reproducing sexually. Fine, let us study. So diploid organism, okay, like you and me, we are diploid organism. So in a diploid organism, we have a pair of chromosomes having genes at the same loci. Okay, so we have a pair of chromosomes, one from each parent, and on these chromosomes, when you look at the genes, let us say the gene for your eye color, you have one gene for eye color on one chromosome, and at the same position, at the same location, which is called loci, you have a gene for your eye color from your father, right? So this 2n condition or diploid condition is shown like this. These are called homologous chromosomes. Okay, homologous pair of chromosomes. It means these are same chromosomes. It could be chromosome pair number 1, it could be pair number 2, it could be pair number 22. All of these autosomes, okay, apart from the sex chromosome, which are X and Y, they are different. Okay, but apart from those, the 22 pairs that you have in your body are all homologous chromosomes like this. Now consider this. A guy, this man, he has 23 pair of chromosomes, which means in total 46. Then a lady, she also has 23 pair of chromosomes, which is 46 chromosomes in total. Because we are humans, the number of chromosomes remain the same. Now what happens if they reproduce and there is no change? Okay, you are giving 46 chromosome from the man and 46 chromosome from the woman. You will end up with total 46 pair or 92 chromosomes, which is actually the scenario in case of this one type of a rat. Okay, this rat has 46 pair of chromosomes, 46 pair of chromosomes, or total 92 chromosomes. So to maintain the species, okay, that every species has their own set or number of chromosomes. For example, we have 23 pair. Same with chimpanzees have 24 pair. So this number is very much specific and very much set. Okay, you cannot change that number. So to make sure that the child is also having 46 chromosomes or 23 pair, there should be some division, such division that the total number of chromosomes coming from the father or from the man are 23, and same case with the woman also. Okay, 23 single chromosomes and 23 single chromosomes from the father. Then only the baby will have 23 pair or total 46 chromosomes. So this means to maintain the integrity of the species and to maintain the number of chromosomes, meiosis is important. In meiosis or meiosis, however you want to call it, in meiosis, you have recombination of genes that occurs, which means if this is your chromosomes, you got chromosome from your mother, and this is the chromosome from your father, then there will be a sort of exchange, okay, between the two. So let us say if here there was a gene for eye color, let us say blue, and on the other hand you had eye color brown. So here there will be exchange of such genes, okay, exchange of parts of the chromosome, and this will lead to the baby getting a mixed chromosome. Clear? This is why you are different from your mother and father, and you are also different from your siblings. Okay, you are not the same as your sister or your brother. The reason is there have been different kinds of such genetic recombination occurring in the body. Fine. The term meiosis was proposed by Farmer and Moore. This specialized cell division reduces the chromosome number by half. Okay, so it is reducing the chromosome number by half, resulting into haploid daughter cells. These haploid daughter cells make gametes. Fine. So here you have that one cell will divide into two. Those two cells will divide and give you four cells. So in the end, you are getting four different cells. It represents the haploid phase in the life cycle. So if we talk about, let us say, human life cycle, then in testis and ovary, which are the gonads, meiosis happens. Okay, meiosis takes place, and during this meiosis, the haploid egg and the haploid sperm are produced. As soon as the egg and the sperm fertilize, it again becomes diploid, right? So from zygote till adult, uh, we are in diploid phase of life cycle, but only after meiosis, the egg and the sperm, they are showing the haploid part of our life cycle. Clear? Now, meiosis involves two cycles of nuclear and cell division. So there will be two cycles, two times the nucleus will also divide and two times the cell will also divide. Each cycle is called meiosis or meiosis 1 and meiosis 2. However, it is important to remember that the cell DNA will duplicate only once. So the whole DNA of a cell will be duplicated only once. Okay, it will not duplicate after meiosis 1 again. Interphase is same as mitosis, which we have already studied. Meiosis 1 is heterotypic division. Hetero meaning different. Why is it different? Because both the cells, okay, both the daughter cells, they will be getting only half the amount of original DNA. Okay, so it is also called reductional division. There is reduction in the number of chromosomes. It leads to reduction in chromosome number by segregation. Segregation is separation. Okay, separation of the homologous chromosomes. And in meiosis 2, it is same as mitosis. Okay, it is called homotypic because both the cells will get equal amount of DNA or equational division. Fine. There is no change in chromosome number and the sister chromatids are separated here. See, this is the major difference. In meiosis 1, the homologous chromosomes are getting separated, while in meiosis 2, the sister chromatids are getting separated. This leads to generation of 4 haploid cells in total. You will understand this better when we start discussing it in detail. Let us begin with meiosis 1. Meiosis 1, again it has all those 4 phases: prophase, metaphase, anaphase and telophase. Prophase 1 is special. It is very much different from your prophase of mitosis. It is divided into these 5 sub-stages. Okay, these sub-stages are called leptotene, zygotene, pachytene, diplotene and diakinesis. We will go through all of these one by one. I normally have a hard time remembering the names. I know the process what is happening, but I forget the name for each of them. So what I do is I have made this mnemonic, okay, this thing to remember it easily. I don't know if it may help or it may not help for you. You can also make your own if you wish. I remember this sequence of leptotene, zygotene, pachytene, diplotene and diakinesis by the statement Le zyada pachan di dawa. Okay, just think of a Sardarji, okay, and he's talking to you in Punjabi. What is he saying? Le zyada pachan di dawa. In Punjabi, you call it di instead of ki. Okay, pachan ki dawa. Instead, you say pachan di dawa. So here he is saying, this Punjabi guy is saying le for leptotene, zyada for zygotene, right? Zya. Pachan, because it is similar to this pachytene, right? Pachan di dawa. Clear? The diplotin and dikinesis differ by this A, right? The di is same. So I thought of this as dawa. Fine. So le zyada pachan di dawa. Let us see the lay first, or leptotine. Leptotine in some books or in some parts also it is mentioned as bouquet phase or bouquet stage. Here the chromosome start to show, okay, the chromatin condense as in prophase 1, in prophase of mitosis also we had seen that the chromosomes start to condense. So here they are very thin and long. They have not condensed fully. They are still very long and they are in the process of condensation. The chromosomes gradually visible under the light microscope. They become visible under the light microscope. This is leptotene. Then you have zygotine or synaptotene. Now here what will happen is, remember I told you we have 23 pair. So each pair, okay, the number 1 chromosome from your mother and the number 1 chromosome from your father will come together. So these different number of chromosomes, they will find their pair and they will start getting attached. This pairing of homologous chromosomes is called synapsis. Okay, can you see this must be, let us say, chromosome number 1 from your mother, and beneath it is your chromosome number 1 from your father. There is this white colored thing you see, that is actually protein. Okay, and this protein will be binding the two chromosomes, the two homologous chromosomes together. So this is called the synaptonemal complex. Okay, the white colored protein that you see is called synaptonemal complex, and synapsis is basically the two chromosomes coming or getting fixed one upon the other. Okay, the pair is called bivalent. Okay, because they have these two chromatids, so they are called bivalent, or they are also known as tetrads because in total they have 4 chromatids. Clear? So bivalent because you have a homologous pair. Okay, the pair is called bi. Bi means two chromosomes of the same type, hence bivalent. Or you have 4 chromatids in total, 2 from your mother, 2 from your father. So they are also called tetrads. So these tetrads, they are visible as just one chromosome when you look under a microscope. It is difficult to differentiate between these two chromosomes because they are almost sewed in together. Okay, they are sewed in, सिलाई हो गई है दोनों के बीच में ऐसे. So bivalent means a pair of homologous chromosome, and tetrad means in total you have 4 chromatids. This much is clear. Just remember these terminologies, okay? Do not get confused. Then comes pachan or pachytene. In pachytene, the meaning of the word means thick thread. Here, the recombination occurs. How I used to teach it on board is, or how I want you to remember it is, I used to say कि गब्बर आएगा, and Gabbar will be like, ये हाथ मुझे दे दे ठाकुर. And then there is exchange of hands or legs between Thakur and Gabbar. Same way here also you have a pair. Let us say this guy is Gabbar and this guy is Thakur. So Gabbar is saying, Tera haath mujhe de de Thakur. And then they are actually exchanging the hands or the legs. Fine. So this part takes place in pachytene. There is recombination. Recombination is you are combining the two things. You are combining the two chromosomes. Occurs between non-sister chromatids. See, sister chromatid meaning from the same DNA you have duplicated. Okay, so if this is the first chromosome, okay, from your mother, it will have one original chromatid and one sister chromatid. So both of them are sister chromatid for each other. Same way from your father also you will have sister chromatids. But if you take one chromatid from your father and one from your mother, then these both are called non-sister chromatids. Okay, one here is light blue and one here is dark blue. So if you take such mixy, it will be called non-sister chromatid. Fine. So there is crossing over or recombination between non-sister chromatids. Always remember this. The point crossing over is the process. Okay, how you cross your legs or how you cross your arms. So crossing over, it is a process. But the place or the point where there will be crossing over is called chiasma. Okay, the point where there is crossing over is called chiasma. Remember this word, it will also come when you study the brain, okay, structures of the brain. There is one optic chiasma, right? So chiasma basically means a cross. So where is this cross taking place? That place is called chiasma. Okay, can you see here the Gabbar and the Thakur? They are exchanging their own um, appendages, okay, their arms. Crossing over is mediated by an enzyme called recombinase. It will not occur on its own. There is an enzyme which is helping the process called recombinase. The chromosomes still remain linked to one another at the site of chiasma. So wherever there was this crossing over, at those points, both of these chromosomes are still attached. Okay, they remain attached at the site of chiasma. Only the part which I have highlighted in the red are included in the patchy team. Okay, this part is not included in patchy team. This is just like a homework. I want you to pause here and look at it, fine, and find out how many crossing over have occurred. Okay, how many chiasma are there. I want you to figure it out. Pause here and figure it out on your own. Okay, and also I just want you to see that how many recombination can actually take place between two homologous chromosomes. Okay, and how it is important. Fine. So after pachytene, Pachan D, right? So D is for diplotin. Diplotin is the phase where the synaptonymal complex dissolves. Remember that cilia in between the two chromosomes, that synaptonymal complex was there. Now, like a zipper, this cilia will be broken down. Okay, this sewing will unravel. So only at the point of chiasma where there was crossing over, the chromosomes will remain attached. Otherwise, they will detach from each other uh, from all the other places. Okay, so this synaptonymal complex will dissolve. The homologous chromosome, they start separating. They look like they are separating, but their chiasma are still attached, like here and here. Diplotin may last up to months or years in oocytes of some vertebrates. Okay, in vertebrates, the oocytes they get frozen. Okay, they will develop during embryogenesis, and then once the baby is born, in that baby, in her ovary, the oocytes will freeze, and they will only again become active once the baby reaches puberty, right? So at what stage does this stop? So and diplotin stage, the oocytes would have stopped. Okay, the last phase in prophase is called diakinesis or dawa, which I told you, right? ले ज़्यादा पाचन दी दवा. So now we are talking about the last phase, diakinesis. In this phase, the chiasma finally reaches the tip of the chromatid. Okay, it will move towards one side. Okay, the crossing over, it will continue to move, and the chromosomes will keep on separating, and it will reach almost the tip of the chromosomes. Can you see here, right? The chromosomes are fully condensed. Here you will find very dense, very condensed chromosomes, and the meiotic spindle gets attached. Okay, the spindle fibers get attached to the different sides of the centromere. Centrioles start moving towards the opposite poles, and the nucleolus as well as nuclear envelope disappears. So at the end of diakinesis, you see that the nuclear membrane or the nuclear envelope and the nucleolus will dissolve. After prophase 1, after all those 5 stages comes metaphase. Metaphase is where the homologous chromosomes arrange at the equator. If you remember in mitosis, we saw 46 chromosomes aligning at the center. While in case of metaphase 1 in meiosis, they will align in form of pair. Okay, the homologous chromosomes will align in pair. So one chromosome will be towards one side and the other chromosome will be towards other side. Clear? This is how they have aligned. The microtubules coming out from the centrioles will attach to only one chromosome, and the same thing on the other side. So this blue colored chromosome will only be attached to this one pole, and the red colored chromosome will be attached to the one pole. This is how, see, slowly the whole pair is getting ready to be at the center of the plate. Okay, and see how the whole pair is aligning. Okay, not single chromosomes, the pair. Anaphase 1, here the kinetochore fibers, okay, spindle fibers, they shorten, they pull the homologous chromosomes apart, and now the single chromosomes from the pair, they will start moving towards the opposite pole. The sister chromatids remain attached at the centromeres, and the division of centromere is absent. There will be no division of centromere as we saw in mitosis. Remember, there was cohesin protein, and we were lengthwise cutting the chromosome and separating the sister chromatid. However, here the sister chromatids remain together, but the homologous chromosomes, they have separated. One side Gabbar is moving, and on the other side Thakur has moved, okay, retreated. See here, each from the pair, one chromosome will be moving on the each side. Then you have telophase 1 and cytokinesis 1. In telophase 1, the nuclear membrane and the nucleolus, they reappear. Fine, here I'm talking about. This is still one cell, so you have to divide it. So cytokinesis follows telophase 1, where the diploid cell, this is still a diploid cell, but when it divides, it will now give rise to two haploid daughter cells. Haploid because one only has the chromosomes from on one side, okay, one pair of homologous chromosome, while the other one has only one of the pair of the homologous chromosome. Clear? So on the one hand you only have Gabbars, and on the other hand you only have Thakurs in my example. Fine. So these daughter cells, they will be haploid. There will be no pair which we normally see in mitosis. This is called dyad of cell. There is interkinesis, like intermission, right? Interval. So there is interkinesis between meiosis 1 and meiosis 2. There is a small gap between meiosis 1 and meiosis 2. In between or in this gap, there is no DNA replication. Okay, always remember this. There will be no DNA replication. Now we talk about meiosis 2. Meiosis 2 is the same as mitosis. Okay, everything is same as mitosis. It resembles mitosis. It is equational division. In meiosis 2, in prophase 1, so at the end of meiosis 1, you now have two haploid cells, right? So these are the two haploid cells, N and N. They are undergoing prophase. In prophase, the chromosomes condense, the nuclear envelope and nucleolus disappear, and the centrioles, they start moving towards the poles. Okay, the centrioles have started moving the opposite direction. In metaphase, again, all the chromosomes, how we had discussed in mitosis, same way. The chromosomes, they align in the central plate or the equatorial plate or metaphase plate. Fine. And if you talk about the chromosome, on the both sides of a chromosome, there is presence of kinetochore, right? The disc-like structure. So from each side, there will be a microtubule getting attached on the same chromosome. Okay, so this centromere will be attached to one pole on the one side and the other pole on the other side. I hope it is clear till here. After metaphase comes anaphase. Anaphase, there will be separation of the sister chromatids. Okay, you can see here, this is the metaphase 2. In the both cells, both haploid cells, the centromere is attached to both the sides of the pole, okay, through microtubules, kinetochore microtubules. When the kinetochore microtubules pull, the sister chromatids will start separating. Just remember the anaphase promoting complex and the enzyme we called separase, right? So it will separate the sister chromatids. Then there will be cytokinesis too after telophase. Okay, telophase is the chromosomes, they start decondensing and get enclosed in a nuclear envelope. Here, as you're seeing, this is anaphase and last one is the telophase. After telophase, there will also be cytokinesis, which will divide the cell into two parts. So here we started with two haploid cells. We are ending up with four haploid cells, all of N type. Clear? And if you look at it carefully, you will see that the DNA in all of them is different. Fine. So you can see a big this light blue colored chromosome and a light blue colored chromosome. Then in this one you have the light blue colored chromosome which has some part of the dark blue. Opposite case here and here. So all of these haploid cells will be very different from the original mother cell. Clear? This is the speciality of meiosis. Just to summarize, in meiosis, you have prophase 1 where the homologous chromosomes align, they do the recombination. Then you have metaphase 1 where they align at the metaphase plate. Then anaphase 1 where the homologous chromosomes separate, and telophase 1, the nuclear envelope develops, and cytokinesis 1, which gives rise to two haploid cells. Follows by prophase 2, metaphase 2 here, anaphase 2 where the sister chromatids separate, and then you have telophase 2 where the nuclear membrane appears, and cytokinesis 2. Just observe this whole process once very silently. Okay, I will just keep on saying which phase it is. This is prophase 1, metaphase 1, anaphase 1, telophase 1, cytokinesis 1, prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis 2. What is the significance of meiosis? It is a mechanism by which conservation of specific chromosome number of each species is achieved. As I just told you that for a species, the chromosome number is very specific. As for us, it is 23 pair. So this 23 pair, the number is conserved because of meiosis. Second thing, it increases the genetic variability. Remember variations? So these variations can occur because of two things. It could either be because of genetic recombination or because of mutation. So mostly it is because of genetic recombination in sexually reproducing organism, and this variation is the reason why you are different from your siblings and you are different from your mother and father. Okay, they are important for process of evolution as well. And this variation is the reason why the cows, they have different coats, or humans, they have different colored hair and eyes. Fine. Now this, I want you to do your homework. I want you to divide your page. Okay, if you have a full-scape notebook, A4 size, divide the page in two parts and write down the differences between mitosis and meiosis. I will just discuss very briefly and very fast, but this should be your homework. Do it very well. Okay, it is for your own good, so that next time when you have to revise, it will be easier. Mitosis is division seen in somatic cells, diploid somatic cells in animals, and diploid as well as haploid cells in plants. While meiosis is seen in reproductive cells and the cell has to be diploid. Both haploid and diploid and haploid cells show mitosis, while in meiosis only diploid cells show meiosis. Mitosis is single division, while meiosis has two division, right? Meiosis 1 and meiosis 2. Mitosis produces 2 cells, meiosis produces 4 cells. Mitosis does not introduce variation. It is specifically to keep that identical type of cell. Okay, while meiosis is to introduce variation because of recombination. The number of chromosomes are same as mother cell in case of mitosis, while the number of chromosomes are half the number of mother cell. Okay. Mitosis is required for growth, repair, and healing, while meiosis is required for sexual reproduction. Now, if we discuss prophase in mitosis and meiosis, in mitosis it is much simpler, while in meiosis the prophase 1 is very, very complicated. We saw the 5 different stages. Okay, ले ज़्यादा पाचन की दवा, पाचन दी दवा. Fine. Here, the bouquet stage and the synapses and all of that is absent. The homologous chromosomes never even meet each other in mitosis. While here, the synapses occurs and the homologous chromosomes, they do get sewn together. Fine. Then in mitosis, the crossing over and chiasma is not seen. In meiosis, crossing over is seen. If we talk about metaphase, all the chromosomes, okay, all the 46, they line at the metaphase plate. While in metaphase 1 of meiosis, the homologous chromosomes, they line up in 2 rows. Okay, there will be 2 lines of chromosomes, and in metaphase 2, there will be a single line again. Anaphase, the sister chromatids get separated in case of mitosis, while in meiosis, the anaphase 1, the homologous chromosomes get separated, and in anaphase 2, the sister chromatids get separated. Again, these are the same things, just a little bit more in detail, but I want you to do it on your own. Fine. And that is all about this chapter. Cytokinesis also, it takes place in mitosis. It may not take place in meiosis, okay? That is in especially in plant cells. Only after all the 4 nucleus have generated, then only the cells will divide. Fine. And as we already discussed, in mitosis, daughter cells and mother cells are the same. In meiosis, the daughter cells are different from the mother. Here we end the chapter. It is a very small chapter, but very, very important. Okay, this unit itself, which includes cell cycle and division, as well as biomolecules, it carries a lot of marks, okay, a lot of weightage. So it is important to remember again, because when you will study human reproduction, you will have to study gametogenesis. If you know meiosis, you will be able to study that well. Again, when you talk about reproduction in different organisms, which you have studied in 12th standard, it also consists of mitosis and meiosis, okay, description somewhere or the other. So if you know the difference, then you will be able to very well study the asexual reproduction and sexual reproduction. Fine. Also, because we talked about the variation and genetic recombination, it is important when we will discuss evolution. And finally, I also want to say that the questions may come which are mostly connecting few chapters. Like they may ask you about pollen grain, how many pollen grains, or how many pollen grains can be made from these many cells, right? And such kind of examples may come. So if you know how many cells how many this cells you get after meiosis, how many you get after mitosis, in meiosis also again how many number of chromosomes are present in each cell, this will help you in answering all such kind of questions. So study it well. It may look small, it only has 2 parts, mitosis and meiosis, but it is still very, very much important. Okay, I'll see you next time. Until then, have fun.